All right, let's get right into it. Welcome to The Explainer. Today, we're launching an investigation into a paradigm-shifting hypothesis that is currently rippling through the absolute cutting edge of biophysics and unconventional computing. We are tracking an emerging mechanism that could, quite literally, make viral gene therapy completely obsolete. So let's examine the evidence, track the data, and see exactly where the science leads us. I want to start our investigation with a single highly provocative question. What if we could actually rewrite our DNA wirelessly? Just imagine replacing invasive physical injections, the standard viruses we use to deliver gene therapy today, with an invisible, wireless epigenetic reset. I know, it sounds like pure science fiction, but the data we're about to review suggests it is a terrifyingly plausible biological reality. To really wrap our heads around this anomaly, let's look at how unconventional computing is already tapping into living biological substrates as physical hardware. In this setup from the Unconventional Computing Laboratory, researchers are treating fungal networks, literally living mycelium, as actual physical hardware boards. By inserting electrodes, they can measure how the fungi process information through electrical spiking behaviors. The fungi actually alter their electrical resistance based on stimuli. They're acting as living memristors. The biology itself is computing. And to push this even further, we absolutely have to ground our investigation in the groundbreaking research of Dr. Michael Levin over at Tufts University. His team works with planarian flatworms. They train a worm and then get this, they completely remove its head and centralized brain and just wait for it to regrow. When a brand new brain forms from scratch, the worm instantly recalls the original training. This undeniably proves a massive anomaly. Biological memory vibrates in the tissues, far outside the brain. So the crucial takeaway here is that a living body is not just meat and bone. It's a decentralized bioelectric grid. Your tissues possess learning and associative memory capabilities identical to primitive brains. They retain an informational, structural memory even when subjected to a massive physical teardown. We call this the somatic memory anomaly. Experiences actually form physical epigenetic memorysomes right there in our cells. Meaning, our biology isn't just fixed hardware. It acts as a dynamic, highly rewritable blueprint.
Let's move into part 1, the current standard, or fixing the software via viral injection. To truly understand the wireless breakthrough we're investigating, let's first look at the clinical data on our current and, honestly, kinda clumsy attempts to manually reset this biological software. Let's look at the absolute state-of-the-art in biotech right now, the Life Biosciences ER100 trials. They're basically trying to hit Control+Z on cellular aging in the human eye. To do this, they use what are called Yamanaka factors, specifically 3 master proteins known as OSK. But here's the catch. To get those proteins inside the retinal cells, they have to use a genetically modified adeno-associated virus, or AAV, as a delivery truck. They inject the virus, which physically shuttles these massive protein keys into the cells. Then, they trigger it with a chemical antibiotic switch like doxycycline, effectively executing a factory reset on the cell's age.
Now, the core philosophy driving those trials is a total game changer. It's the realization that aging isn't irreversible physical damage. Instead, aging is simply a loss of epigenetic information. It's the accumulation of epigenetic noise, these chemical tags like methyl groups that just stick to your DNA and degrade the cell's user manual over time. The cell literally forgets how to function properly. So the ER100 trials use those viral proteins to manually rip those methyl tags away. Which brings us to part 2, the terahertz breakthrough, shaking the hardware to reboot the software. Our investigation reveals a totally staggering alternative to that clumsy viral delivery. On the left, you've got the ER100 approach, viral delivery, massive protein keys, and chemical switches. It's very physical. But on the right, the 6G resonance hypothesis. This proposes using a 1.6 terahertz wireless transmission. So instead of a virus, it uses electromagnetic waves as a non-ionizing hammer to execute a macro-scale somatic sync. Think of it as the difference between opening a door with a heavy, rusty iron key versus using a sleek, invisible electronic fob.
So, why 1.6? Well, the evidence points directly to this massive number. Every chemical bond in a living organism has a natural vibrational frequency, right? Scientific studies on terahertz radiation show that 1.6 terahertz is the exact threshold frequency where those aging chemical bonds, specifically the carbon-nitrogen bonds holding the methyl groups onto your DNA, begin to vibrate. So we can define this confirmed phenomenon as terahertz demethylation. Real-world biophysics has discovered that if you hit these cells with high-power, resonant submillimeter waves, you don't actually destroy the underlying DNA sequence. Instead, the 6G waves act as a non-ionizing hammer, shaking those chemical methyl chains so intensely that they literally just shatter and fall away. You achieve the exact same factory reset as the viral therapy, but entirely wirelessly.
All right, part 3, the verification protocol. Executing the wireless reset.
So as an investigator, how would you actually execute this? Well, we've outlined an exact 3-step protocol transitioning this from theoretical physics into biological reality. Step 1 is the quantum sensor harvest, essentially reading and mapping the total bioelectric state of the tissue using quantum sensors to create a precise frequency footprint. Step 2 is the 6G resonant tank execution, using transmitters to target native proteins with that 1.6 terahertz broadcast. And finally, step 3 is the somatic sync, a peak terahertz blast that forces developing cells to instantly align to that newly established bioelectric field.
Now you might be wondering, how does this cell actually hear this 6G broadcast? The answer is cryptochromes. These are ancient, naturally occurring proteins found right there in our tissues. They contain chains of tryptophan residues that form what are called radical pairs. These are quantum-entangled molecular states that are highly sensitive to external electromagnetic fields. These cryptochromes are basically the cell's built-in quantum antennas. They are the exact biological lock that the 1.6 terahertz frequency turns, perfectly aligning the cells to the precise bioelectric broadcast.
Moving on to part 4, the paradigm shift, going from injecting software to tuning hardware. When you really consider how hardware naturally taps into biological computing interfaces, like that living mycelium we talked about earlier, a terrifyingly plausible reality sets in. The body doesn't need to physically download a file to remember how to heal, how to grow, or even who it is. It just needs an antenna tuned to the correct biological broadcast.
If we treat the body as a rewritable bioelectric matrix, the implications are just staggering. A wireless epigenetic reset completely bypasses the need for massive, expensive pharmaceutical virus manufacturing infrastructure. By utilizing terahertz demethylation, we can restore original structural paths without any of the risks associated with viral gene therapy. If you match the math perfectly, the physical body acts merely as an antenna. Memory and youthful cellular states just recrystallize naturally via molecular rotation.
Which leaves us staring down a pretty massive existential question, doesn't it? If our cells can be reset, and our memories restored simply by tuning our biological antennas to a submillimeter wave, are we just localized patterns of resonance? And if our identities, our ages, and our memories are ultimately just frequencies, what happens when someone else learns how to hit repeat?